Hello, everyone. Today, it's my pleasure to introduce the paper submitted to iMeta. The title is scRiskCell, a single-cell framework for quantifying islet risk cells and their adaptive dynamics in type 2 diabetes. Type 2 diabetes is a common progressive disease, typically developing from normal glucose levels to prediabetes and eventually to diabetes. However, our understanding of its progression mechanisms remains limited. As the central target organ in diabetes, the pancreas exhibits highly heterogeneous cellular composition. Studies have revealed the coexistence of healthy and diseased cells in diabetic pancreases, but how their dynamic changes contribute to disease progression remains unclear. To address these challenges, we developed scRiskCell, a machine learning-based computational framework that identifies cell type-specific risk populations, including rare islets epsilon cells, decoding their mechanistic contributions to the progression of diabetes. We systematically analyzed nearly 500,000 islet single cells from 106 donors across different disease stages, constructing a comprehensive human islet cell heterogeneity atlas during diabetes progression. Our analysis identified 11 major cell types, including rare epsilon cells exhibiting unique growth hormone secretion and metabolic signatures. Application of scRiskCell revealed a progressive increase in beta risk cells during diabetes progression, with significant differences across clinical groups. The risk cell proportion signature alone achieved accurate disease state classification, with robust validation in independent cohorts. Moreover, scRisk generalized to other islet cells, including rare epsilon cells, successfully identifying disease-associated, cell type-specific risk populations. To assess clinical relevance, we examined associations between beta risk cell proportion and clinical traits. Risk cell proportion was lower in younger individuals and showed an increasing trend with age, although it did not reach statistical significance. No significant differences were observed between sexes. Obese individuals had a higher beta risk cell ratio, with a significant positive correlation between BMI and risk cell proportion. Hemoglobin A1C levels positively correlated with risk cells, while C-peptide showed a negative correlation, consistent with diabetes progression and beta cell dysfunction. Transcriptomic comparison of risk versus non-risk cells enables high-sensitivity detection of molecular changes associated with diabetes, especially in beta cells, where 2,734 differentially expressed genes were identified, far exceeding than those detected using donor labels. Other cell types showed similarities. Gene regulatory network analysis revealed that risk beta cell networks are primarily governed by transcription factors related to endoplasmic reticulum stress and apoptosis. These hub factors drive target genes involved in endoplasmic reticulum stress, oxidative phosphorylation, autophagy, and apoptosis processes. Heterogeneity analysis of beta cells revealed that risk cells exhibit dynamic clustering patterns during disease progression. Risk cells initially clustered in beta subgroups 1 and 8 during the early stage, then expanded to subgroups 3, 5, 6, and 7 in the disease stage. The early active subgroups 1 and 8 were primarily involved in glucose metabolism and protein secretion, suggesting an adaptive response. In contrast, disease-associated subgroups 3, 5, 6, 7 showed enrichment of genes related to ribosomal function, protein folding, autophagy, and endoplasmic reticulum stress, reflecting massive dysfunction of fundamental cellular functions. Summary: we developed scRiskCell, a computational framework that identifies disease-perturbed risk cells from large-scale human islet single-cell profiles. The risk cell signatures reveal diabetes progression trajectories and demonstrate robust generalizability across independent cohorts. Furthermore, these signatures cover key regulatory programs and dynamic perturbations underlying islet cell dysfunction during disease progression, providing new insights for early diabetes warning and mechanistic research.